I'm a PhD at Yale University and I study human aging. Specifically, I build biomarkers of aging to quantify how different organs in our body are aging from a single DNA methylation blood draw. And then we can really use these biomarkers to potentially build new medications that might slow down aging. And that's kind of what my research is really on.
And why is this important?
Well, the reason why this is important on a broader scale is that we know that we have an aging population. We would like that hopefully we can build medications to allow people to live longer, healthier lifespans. Uh, but on a more simplistic level, to be able to do this, we first need to be able to essentially build biomarkers that can measure how quickly a person is aging. Uh, but more importantly, if we actually have to run clinical trials, to run a clinical trial for aging, you cannot actually run a clinical trial for aging at this point because that would effectively take 30 years because the endpoint is mortality or death. So to counteract that, you need to have proxy biomarkers. Proxy biomarkers that can tell you that, oh, whether an intervention is working or not. And that's what really I'm trying to do. Build these biomarkers that can really allow us to tell us whether an intervention is working or not. And that would allow us to essentially build these interventions and clinical trials that can run very, very quickly and in a very cost effective manner. So instead of running a clinical trial for 30 years now, we can run a clinical trial maybe in 2 years at a much cheaper price. And that would hopefully get us an intervention much quicker.
And how far off do you believe we are from such a set of biomarkers that would enable fast clinical trials on aging specifically?
I think we are not too far off, in my opinion. So we already are seeing a lot of aging biomarkers from proteomics, DNA methylation, metabolomics that are performing really well in sort of what we call as three categories. One is prognosis, which is the ability to predict future diseases and future mortality. The second is responsiveness, the ability to respond to intervention. And the third is reliability. That means that they do not significantly change, say, between one test to another. And that's the exciting part. But there is scope for improvement. So we really need to potentially even improve these biomarkers further before they can move into trials. The second thing is we also need to validate which of these biomarkers go into trials. So here's the idea that before even we move them into a clinical trial, we need to run what we call as a surrogate endpoint trial. Now, this might sound a little complicated, but what we really need to do is run a clinical trial for a biomarker. We need to see that whether one of these biomarkers is actually predictive of change, of mortality, of diseases, and response in a clinical trial setting. So, for example, we take a biomarker for aging, right? Give it in a very well known intervention like exercise, which we know will hopefully slow down aging, right? Or calorie restriction, which we know will hopefully slow down aging, right? And then say, does it actually track with other outcomes of mortality and disease, disease occurrences and frailty, so on and so forth? And if it does, then we have a surrogate endpoint or a surrogate biomarker, which is great, perfect. And now we can use this biomarker in actually clinical trials and give it to patients all across the globe to track their health, and which would hopefully transform preventative medicine. Because now you essentially have an aging biomarker that works for all practical purposes.
What has you most excited about the field over the next 5 to 10 years?
I think what's most exciting to me over the next 5 to 10 years is the ability to do a lot of these analysis. We are generating so much data through these longitudinal cohort studies, through these small interventional studies, that we can really now look and do systematic analysis around these biomarkers, around these interventions, and how they are really changing human lifespan and health span. So we can really start drilling down on what's working and what's not working. So I do believe we are still like maybe 10 years away from getting our first gerotherapeutic. But at the same time, we are really moving forward towards at, I would say, light speed. Because at this point, we really have tons of data to really start validating all these biomarkers and interventions that we have had on the back burners for such a long time. And that's the really exciting stuff, in my opinion.
Well, given that we're at the Biomarkers of Aging Conference, if you had to put all your chips on a single biomarker right now that you think has the best performance in terms of measuring aging or predicting the speed of aging, whether people are aging gracefully or not, what would it be?
I don't think I can put a bet on like a single biomarker. I think that would be a disservice in a way. But I would say that there are definitely sort of platforms of omics that I feel are more powerful than others at this point, looking at the data. So something like DNA methylation, right now, we have been able to systematically see that it's prognostic, it seems to be responsive and reliable. We haven't done the same analysis for proteomics, we haven't done the same systematic analysis for metabolomics and for clinical biomarkers, at least in the context of aging. Until we do those systematic analysis, we won't be able to at least say that those platforms or those omic data types are sort of ready for clinical surrogate endpoints. So what I'm saying is that I don't think there's going to be a biomarker, right? I'm guessing there's probably going to be multiple biomarkers. But what we really need to do is validate the platforms first, right? Whether the platforms are ready, and then validate the individual biomarkers. So my bets at this point seem to be on DNA methylation, partly because I work in DNA methylation and that's where I'm biased as well. But that's also because it has probably had the most progress at this point in context of aging biomarkers. And that's I guess the situation we are in.
It's very exciting.
Yeah.
It's great speaking with you.
Thanks for the conversation.